chapter 24, speciation. I'm sorry, I'll start population genetics on the next round. We could do them in either order. So this is a picture of Darwin's finches from the National Museum in Baltimore. So you can see the beak differences, right, of our birds here. So they all look like finches, but you can see that some of them have smaller beaks. Like this guy here is more of an insectivore, where this one here, one of the larger ground finches, it can crush very large, thick seeds that the other birds can't feed on. And so these guys are what are called niche specialists. Their niche is what they eat. And their beaks have been shaped over millions and millions of years to fit a food source. So our selection pressure here is food, and food is what is shaping the phenotypes that you see. So the big question becomes, first and foremost, I keep talking about a species, right? Or these individual organisms that we recognize as entities. So species is the Latin word, or Greek Latin derivative that means kind, right? A kind of, or appearance of. And biologists used a whole bunch of different types of data to categorize an organism into a species. So if you find a new organism out there, say a new spider or a new amphibian or a new plant, you have to figure out if it's truly a new species, and you can do that many different ways. Often, the first thing that we do is catalog really detailed morphology of the organism, and then take a sample so we can do a DNA sequence and then compare that with all the other organisms that we know. And that'll allow us to know if it's a new species or not. The big question comes down is once we kind of, define what a species is, then biologists have to, using evolutionary theory, explain how speciation occurs. That is, how do closely related species, how are they related to each other, and who did they come from, right? Or how does speciation happen? If you have a population of one species, how does it split into two species, is the question. And we're going to go through how we have observed that kind of event and how speciation actually occurs over millions of years. So basically what we're going to do is what we've talked to this point are changes in genotype and phenotype from generation to generation, right? These little meandering changes. What we need to do is use large time frames to accumulate those micro changes so that we can actually produce separation above the species level of organization. And that's what we're going to talk about today. This is a wonderful bird right here. This is the Galapagos cormorant. So cormorants, there are lots of different species of cormorants, different kinds. This one is found exclusively in the Galapagos, and it's one of the only ones that can't fly. So you can see how short its wings are. Probably from a mutation after it landed on the islands that prevents it from flying. And now it breeds and lives exclusively in the Galapagos, and obviously looks different and behaves different than the cormorants on the mainland of South America. We have cormorants that fly through Indiana. Same species that's in South America actually migrates into the United States in the summer and then goes back in the winter. Obviously, it's not this one. This one is isolated because it can't fly, right? It's stuck now in the Galapagos. Fish eaters, they eat fish. So we're going to explain evolutionarily how species originate and how populations evolve. And remember, I've already mentioned that it's populations that evolve, not individuals, right? So it's populations that evolve, not individuals, and genetic variation within that population is really important, right? So how do you define a species? It's probably the worst question somebody can give you biologically, because in reality, it's an artificial thing, right? It's how we define what we see. And we argue a lot, particularly these people who got all this education. We like to argue over, how we define a species. There are about 25, 26 different species definitions now. And different biologists use them in different disciplines. The one that makes the most sense to me, and still does, after all these years, is what's called the biological species concept. That is, if you think two birds that look alike are different species, you simply have them breed, or try to breed, right? And if they can breed and produce viable offspring that can then breed, then they're not different species. If they can't reproduce, or they produce babies that are sterile, or they produce babies that die, then they are different species. So it's all whether you can reproduce or not. And this is a wonderful concept until you find asexual organisms, right? Oh, what about the organisms that don't have sex, right? They just produce copies of themselves. Or gene transfer. There are salamanders that the female breeds with the male and steals genes and then throws away the rest of his genome. How rude is that, right? Especially if you're male, which I am. Fossils, it doesn't help with fossils. And then there are some species that just don't behave or can crossbreed even though they never spend any time with one another. And we treat them as species. So there are other ways to define species when the biological species concept doesn't work. It's the one I usually default back to. But there are, for example, here I'll give you 3 others. I don't give you all 20 of them. There's morphological species concepts where we define things based on what they look like, right? And we do that immediately, automatically. That's kind of how taxonomy works. The biggest problem with this is convergent evolution, right? Remember convergent evolution? When two organisms look alike, but they don't share common ancestry. It's the environment that shaped them, like sharks and dolphins, right? If we put those together as similar species or close to each other, then they're not because the environment shaped them. Ecological species concepts. This is a good one. One of my probably my second favorite. Ecological species concept is the concept behind defining species based on the niche they occupy. An ecologist will tell you that no more than one species can occupy a niche at a time, a niche being a very specific part of the environment. And the species concept is nice with evolution because remember, it's typically the environment that is working on the phenotype to fit a niche, right? So it kind of sits real nicely. And so it is going to be based on something that we call disruptive evolution. And what disruptive evolution is when you have your species, right? One species like this, and most of them look like this, right? And basically the environment is going to shape it so that it looks like this, right? So you went from one hump to 2, and you have now 2 species, right? Or you select for the extremes, and they fill a niche, right? They fill a specific niche. And typically what's really nice about this is when a species like this mockingbird fills its niche, and that mockingbird fills its niche, then they don't interbreed because they spend time in that niche only, right? And so there's no interbreeding, and that is what's really important because that leads to speciation. As you're going to see here in a minute, the first thing we need is reproductive isolation, right? So that they're not interbreeding. Because if something can interbreed continuously, it's not a different species, right? Number 4 is the evolutionary, or what they call the phylogenetic species concept, where they group organisms based on dichotomous branching, or how closely related they are to each other. And we're going to do some phylogeny work as we move through the next few lectures. So here are Galapagos mockingbirds and where they're found on the archipelagos, and they all feed on different materials often, but birds separate themselves a lot based on sexual selection, that is female choice, and each of these have kind of a different song and behavior to attract their mate, and that's what reproductively isolates them. And that happens a lot in birds. The ability to have different song and behaviors that we're going to call a pre-zygotic barrier. So we'll talk about that in a minute. So you remember particularly this morphological concept, or it's really easy to start grouping things that look alike, right? Or not group things that don't look alike and put them in different groups. Here are some good examples of where morphology doesn't work. One of my favorite birds, a beautiful Beautiful birds, about that big. These are called meadowlarks. They have a beautiful song. This is the eastern meadowlark, and this is the western meadowlark. I mean, they look really alike. And sometimes it's almost impossible to tell them apart. So if I took a picture of this bird and took a picture of this bird, I'd be like, I have no idea which one it is until they sing. All right, when they sing, then the eastern song is much different than the western song. And the females of the eastern meadowlark prefer the eastern song, and the western females prefer the western song. So even though they look alike, their behavior and their song isolates them reproductively. And they won't interbreed. So here you got two that look alike, and you might say, oh, that's the same species. And in reality, they're not, right? Down here, these are two caterpillars. Here's a caterpillar, and here's a caterpillar. When these were first found, they were thought to be two different moth species. And in reality, they're the same species. Isn't that crazy? This moth, I mean, I would have never guessed that. And the way they figured this out is by taking the caterpillars, putting them in a tank, and allowing them to, feeding them, allowing them to form a chrysalis, and then when they hatch out, they're the same moth. Their genotypes are often exactly the same. So it's incredible, because you know that genotype determines phenotype, right? That's what I've told you over and over and over again, right? Yes?
How are they the same that it's
That's a great question, and I'm going to tell you how right now. You're good. So it's all based on what they eat, actually. So in this instance here, and this is one of the rare instances, where phenotype is not determined by your genetics, but determined by your environment. And so the moths lay eggs kind of wherever they want. The caterpillars just eat, right? And so these guys, their host plant is the oak tree. And sometimes the eggs are laid on flowers, the oak flower, and sometimes they're laid on the leaf, which is behind here. The oak flower is small and this colored, and when the eggs are laid on oak flowers, the caterpillars tend to eat the oak flowers, because they eat where they're, eat whatever they're laid on. And the chemical cues in the flower cause them to take on this morphology by controlling their development. And so they take on this phenotype and look exactly like the flower, so that when they're sitting on the flower, they're not going to be eaten, right? If you're laid on the green leaves, then you take on this phenotype, because the chemical cues in the green leaves are different, chlorophyll being one of the main ones, than the flower, the chemicals that the caterpillar is exposed to in the flower, and they look like a twig. And so it's not their genotype that's controlling these phenotypes or the morphology, it's the environment they're in and what they eat. And so there are some other insect examples like that. So these are really neat, and this is why biology is, really interesting, because you can always find exceptions to the rule. Isn't it nice to find exceptions? And this is a big one. So does that answer your question? So what is the first step of speciation? So how do I become two new species? The first thing I have to do is become reproductively isolated and then stay that way for a long time. Typically a long time. So how do I do that, right? So how do I produce this barrier that prevents me from reproducing? Because that's really, if we're thinking about the biological concept, it's probably pretty safe, right? If species practice sex, which most species do, then if we bring them together and they can't produce babies that can breed, then we know they're different species. So what are these barriers, and how do they arise, right, is the big question. And so we've come to recognize through observation and testing that there are several types of barriers that separate species. All right, some of these are called prezygotic barriers or mechanisms, and some of them are called postzygotic barriers or mechanisms. So where's this name pre and post come from? So prezygotic means these are going to be mechanisms that arise in organisms that prevent the male and female from actually reaching the point of mating or finding each other. All right, so they never breed. They never release gametes to each other. And so they're called prezygotic because they happen before the zygote, right? You guys remember what the zygote is, right? It's the product of sperm and egg meeting, right? So we're going to prevent the sperm and egg ever from meeting and forming a zygote. There are 5 kinds, and there are 3 types of pre-mating, preventing mating altogether, so that sperm and egg will never come together, and 2 post-matings where, the sperm and egg did meet, and something happens to the zygote or the embryo afterwards. There are also post-zygotic mechanisms that prevent that embryo from developing right? And so there are 3 kinds of these. And so we're going to talk about the differences between pre-mechanisms and post-zygotic mechanisms. Now this picture up here is what's called an apple maggot fly. Isn't he cool looking? So it looks like a spider, doesn't it? You guys see the jumping spider? So these are the wings. How cool are the wings? The wings look like somebody painted legs on them. That's pretty cool. And so you got the body. This is the head up here. These are the eyes. And so it looks like a spider so that predators leave it alone because it's a spider. Spider will eat other insects. And so that's a nice little trick. It's called mimicry, mimicking spiders. These guys lay their eggs on apples, which they're pretty interesting. I'll talk about them later. So let's talk about these prezygotic mechanisms that prevent viable offspring. So there are 3 that are called pre-mating. So 3 pre-mating prezygotic mechanisms. They are habitat isolation, temporal isolation, and behavioral isolation. All of these prevent the male and female from finding each other. Okay? So what's the difference between them? Habitat isolation simply means that 2 species prefer pretty robustly different habitats from one another. And there are 2, I gave you 2 examples of many. So here are 2 species of closely related cricket. They're different species, and they're considered different species because they never reproduce. They never cross-reproduce. They never produce what are called hybrids. And the reason for that is because the females of this species prefer grass and cover, and these guys prefer sand, a sandy soil. All right, and they're really adamant about finding the right environment. Crickets are. And so the females of this darker species like to be under things that are found in sandy soil. These guys like to be in brush, grass, or vegetation. And so the males follow the females, right? That's the way it goes. Males follow females, no matter what the species is. And so they never are going to find each other, the males and females, because of where the females' habitat preference is. The same with garter snakes. There are several species of garter snakes that one species loves more terrestrial, drier habitat, and then one loves to be in the water. I mean, that's where it is going to live, is in the water or near the boundary of a pond or a freshwater source. And so they might actually be pretty close to one another, say, within an acre or even hundreds of feet of one another, but they'll never interbreed because of their specific habitat selection and the food they eat. So that's what habitat isolation is preferring different habitats. And so mates don't find each other, keep species separated. Temporal isolation also makes a lot of sense. Temporal isolation is when 2 closely related species that might have the ability to interbreed are actually separated by when they mate. Right? So it's a time thing. So frogs are a great example. So here in Indiana we have wood frogs and leopard frogs and several other frog species, but these are just 2 here. These 2 are spring breeders. The wood frog is the earliest frog to reproduce here in Indiana. Typically, I've seen them, in ponds calling as early as March, even late February. So they start calling and breeding, really early in the spring when there's still even ice, in the water. Where leopard frogs, which will be out when the wood frogs are out. I've actually seen wood frogs calling and leopard frogs in the same pond, hanging out. But the leopard frogs need warmer temperatures. It needs to warm up a little bit before they get in the mood to reproduce. And so they typically reproduce, you can see here at the peak, after the wood frogs are done. So they're reproducing at different times and never, cross interbreed and form a hybrid. Same with skunks. Isn't this a cute skunk here? So it's not striped, it's spotted. So there are spotted skunks. There's an eastern and western species, and their populations overlap where they live a lot, believe it or not. And the reason they don't interbreed is because the eastern species breeds in summer, and the western species breeds in the middle of winter. So really different times of the year when the male and females are ready to breed, and they never crossbreed or interbreed because of this temporal isolation of reproduction. And then the last one's behavioral isolation. You see this a lot in birds. So, and I already given you an example of this, where you got two meadowlarks that look exactly alike, but they have very different songs, and that's what the female listens to. And a lot of males have a routine to courtship behavior, like dancing or moving around. Most of them move really elegantly or really fast, so I picked a blue-footed booby because I could be a blue-footed booby if I had to find a mate. They are very awkward and have a terrible song. And they have big, huge blue feet, and they just show them off to the female very slowly, and then they waddle around, and the female digs the more awkward male, the better, right? So they're fun. Look them up online. They're hilarious to watch do their mating ritual. So now there are other boobies. There are brown-footed boobies and green-footed boobies, and they don't interbreed because they all have different waddle dances and different colored feet, right? And so that keeps them from interbreeding these behavioral isolation mechanisms. And then if you want to watch a bird that has an incredible behavioral isolation pre-mating ritual, look up manakins. Manakins are birds of paradise, and the males like moonwalk and dance, and they're pretty cool. They're fun to watch. They're really loud, and they move really quickly. They're fun. They're a neat bird. So a lot of birds do that. So those are all pre-mating, preventing the male and females from finding one another. Now, if a male and female does find themselves together and ready to mate in their different species, there are two major, what are called post-mating, like, the males and females find each other, they're trying to mate, right? They're trying to mate. And what prevents the actual mating, the release of sperm to the egg, are two mechanisms called mechanical isolation and gametic isolation. So mechanical isolation simply means that the gonads can't find each other, or the mechanism of delivering gametes can't find each other. So here's a great example of this. Snails. A lot of closely related species of snails, when they lay their shell down, some of them are right-handed and some of them are left-handed, and so they spiral differently. And so how snails mate is, if my hands are snails, their heads touch, and then they got shells behind them, right? And then one of the heads goes over the top, and it lines up the gonads. But the problem is, if your shells are twisted the wrong way, the shell gets in the way. And so they can't quite reach each other and line up the gonadopore of the female with the male, so the male can't deliver sperm, and you can see they're too far apart. So they can never get sperm delivered to the female eggs. And that keeps these two species separated from one another, just based on how their shell developed during development. Ducks are like this also. So ducks have a spiral penis. It's actually spiraled. And the female's reproductive tract spirals the same direction, so that two ducks that have different female reproductive cavity to male spiraling can't interbreed. Right? They can't, ejaculate sperm into the female reproductive tract. So that's another example. Birds are good examples of that. The last one is gametic isolation. All right, where, you do breed, the gametes find each other, but the gametes are not compatible. That is, the sperm can't fertilize the egg. This happens a lot in animals that release their gametes into the water column, right? So a lot of aquatic animals do this. So here's a great example. Sea urchins. You might be sitting on a coral reef at the right time of year, and there'll be 20 or 30 species of sea urchins and corals that all reproduce that night. It's the highest tide. It's the night of the highest tide or the largest full moon. And the currents are really strong that night. That's why everybody releases their gametes at the same time so that they mix up really nicely. And so you have these 20 different species of sea urchins. How are they not all the same species if they're just releasing their sperm and egg into the water column, right? Because, the sperm and eggs are going to run into each other. So how do they keep species separated? And it has to do with the proteins on the sperm have to match up with the proteins on the egg. So each species has its own specific proteins that are expressed on the surface of the sperm and egg so that when they meet, only compatible ones stick together. So if there's an egg from one species and you're a sperm from another, you just bounce off of the egg because you can't dock, if you will, right? Or match up proteins on the surfaces of egg and sperm. And so that prevents the gametes from actually allowing fertilization to happen. So those are the pre-zygotic mechanisms. Now, what happens if, we mate, egg and sperm meet, fertilization happens? So what are some mechanisms that prevent the embryo from developing correctly? And there are 3 of these that we have identified in nature. These are called post-zygotic mechanisms. They're called reduced hybrid viability, reduced hybrid fertility, and then hybrid breakdown. A hybrid is when 2 different species meet and form babies. The baby is the hybrid. So reduced hybrid variability simply means that once the zygote starts dividing and forming into either the baby or the larva dies during development. So very few of the hybrid offspring make it past the development point. This happens a lot in amphibians. So a lot of salamanders, for example, breed at the same time and in the same pond, and sometimes the sperm and egg of different species meet, and you actually get an embryo, but the embryo dies during development. Or in the larval stage just doesn't make it. It doesn't survive. It either can't find food or can't compete or whatever it is, and none of them will metamorph into adults. So this is when the hybrid breaks down during development. Reduced hybrid fertility simply means that, hey, you mate, you produce a baby, the baby's robust and vigorous, but is sterile. All right, so the babies grow to adulthood and they make it and they're fine, but they can't reproduce. They don't have any eggs or sperm. And so the best example here is a donkey. When a donkey mates with a horse, and you get this beautiful, robust mule, right, which is a great farm animal, but it can't reproduce. It doesn't have any sperm or eggs. And so obviously this isn't the same species, right? They're actually different species because their babies are not viable and they can't reproduce. The last one is hybrid breakdown, which happens a lot in plants. So, because remember, plants are weird. They don't follow the rules. They can reproduce sometimes when they're different species. Yep. In order to be a species, what? Well, yeah, you need to be able to reproduce. But their babies can't make babies. You have to be able to make babies. Your offspring need to be able to make babies in order to be different, the same species. Sorry.
So are mules a species?
They're just considered a hybrid. They're not considered a new species or a separate species. They're considered a sterile hybrid of this breeding event. They're not considered. They don't have a separate species name.
Okay, so is a hybrid, is that like the baby of like 2 like closely related organisms?
Yes.
Like a monkey?
Yeah, exactly. Yeah, that's what a hybrid is. Yep. Yes.
What about hybrids that like can't reproduce?
Then you would consider them not separate species probably, or they haven't completely evolved separate from one another, right? Where they're a completely new species. Now, that's when the species concept becomes really gray, because you might have a species of snake that's 250 miles away from this species of snake, and they'll never interact, right? And so you could consider them separate species, but when you bring them together in the lab, they make babies, and the babies reproduce. So they're like the same species, right? But they give them subspecies names because they'll never meet, right? So yeah, then it gets gray. Then it's like, well, what do you do with them, right? All right, these three make sense? Well, I haven't done this one yet, have I? These two make sense? Everybody good with those?
So essentially, like all the reduced hybrid varieties, that's like within, and the other one that's like with the hybrid baby itself, and not like the two parents.
Yeah, it's the hybrids. The hybrids don't do well. So, for example, if you were in a pond and you had a long-tailed salamander and a two-line salamander, and they're breeding at the same time, all the two-line salamanders that breed with two-line salamanders will produce larvae that are robust and fine, and so will the long-tails. But if the sperm and egg of those two meet and they start producing offspring, it'll die.
Okay.
That way you keep the species separate. Does that make sense? And then with this one, you produce a hybrid, robust, grows up, just can't breed. Okay? The third one, hybrid breakdown, simply means, and this happens in rice a lot, like the plant rice. You can have two strains of rice which are considered separate species, and you breed them together on purpose, and they produce a hybrid that is robust and it can reproduce. And it does a great job of reproducing. And then the next generation, all of the offspring are either sterile or don't survive or fragile or can't reproduce. And the further you get away from that original mating, the more and more and more the hybrid does poorly. So what that means is the parental generation gives you an F1 that's robust. The F2 is not so robust. The F3 is even less. By F4, you can't get any babies. And so in the long run, they're not the same species. Does that make sense? They're actually separate species. That happens a lot with plants because they can deal with multiple sets of chromosomes. Okay, pre-zygotic, post-zygotic, everybody understand those? So those are the barriers that separate species if they're intact, right? And so those barriers have to arise during evolution of two populations in order for them not to be the same species. So the second major step that causes those barriers, pre or post-zygotic, is genetic divergence, right? And so how do you do that? How do you separate animals for a long enough time that they will genetically diverge? And there are two major ways to do that. And think about it. So if you got this population, and just like right here, you got this population of fish sitting around in this big water source, right? And let's say that all of a sudden there are drought conditions that drop the water column here down to this, right? Now you have two ponds. And let's say it stays like this for 100 million years. That means this population of fish is reproductively isolated from this population of fish, right? And let's just do this. Let's just add a predator on the right side that's not found on the left side. And so that's a different selection pressure, correct? A different selection pressure is going to cause natural selection. And maybe their developmental, I'm just making up things here, but maybe their developmental process speeds up, where their developmental process in the bluefish slows down. So the bluefish are maybe larger or different size. If this were to reflood after 100,000 years, now you have two completely separate species whose hybrids don't survive, right? Either because of the developmental difference or some behavioral difference now, right? Between the two species as far as mating. Maybe it's mate recognition. But now you went from one species, 100 million years of separation, to now two separate species, right? That are separated by one or more of those pre or post zygotic barriers. Does that make sense? The barriers form because of the genetic divergence. Typically, you need lots of time in animals for this to happen. Plants are weird. They can do it in one generation. I mean, it's really weird, and I'll tell you how as we start talking about speciation. Now, there are two ways to do it. The first one is the easiest one. The first one is the one that makes intuitive sense, perfect sense. It's called allopatric speciation. Allo means to separate, patric means your population. So allopatry happens when a barrier, a big, huge geographic barrier forms and separates one population into two or more populations. There are two ways to do this. The first one is called dispersal and colonization. So think about it. You got this population. The population gets bigger and bigger and bigger. It gets really crowded. There are a bunch of them, right? You're always going to have some adventurous individuals that say, hey, I'm leaving. And they leave, right? Or they drift away. Or they really push the boundaries of where the population is found. And if they get separated from the ancestral or original population by a barrier that's big enough that it's hard to make it back, you separate the populations, right? The best example of this is island biogeography, like the Galapagos Islands, right? You have South America here and this huge population of, let's say, cormorants, right? Like this guy, right? But they can fly. And you have a few individuals that like to really push it on the coast because there's food sources. They don't have to compete against everybody in the middle, right? And let's say a big hurricane pushes them to the Galapagos Islands, which they wouldn't have tried to make that trip on their own. It's too far, right? But they get pushed out the sea. They're lost. They fly, and they find the islands way out here, and they settle down and start, and there's food there. So they can settle down, form a population there, and they're not going to try to fly back. It's too far, right? They could make it, some of them, but it would be hard, right? So this is a big enough barrier to separate the original population from the new population, okay? Now, if it stays like that for millions of years, there might be completely different selection pressures here than over here, right? And you're going to get new species, or something might happen like a mutation that leads to this, where the bird can't fly anymore, so it's not going to try to fly back anyway, and it will be stuck here and now become a completely new species with completely different breeding behaviors. If you brought the two back together, they wouldn't interbreed. The other thing that I want you to notice is imagine this huge population as genotypes, right? Maybe you have 30 different genotypes that are found in the original population, and only 4 of those genotypes make it to the island. You've already changed the genetics. Only those 4 are going to be breeding. And so those are the only 4 phenotypes you're going to see, where there are 20 over here. They might be really different types of phenotypes, right? That allow them to exploit a food source on the island that wasn't available on the mainland. So you can start to see you're already changing the genetics and what phenotypes actually are there with colonization and dispersal. Make sense? The second one's called vicariance, and this is when a big, huge geologic barrier divides a population into 2 populations. A volcano, a tsunami, earthquake, changing river, trajectory, the rising of mountain ranges. That's all of a sudden separate. Whoops! I'm throwing things. Separate an ancestral population into 2 new populations. And so now this barrier is so complete that these guys will never interbreed. This has happened a lot during the biogeography and movement of all of the continents and Pangea and mountain ranges that arise. And you can see it pretty easily when you start doing some experimentation. So this makes intuitive sense, right? It's really easy to separate populations this way. Let me give you 2 examples of how this works. So you can go out in nature and find it, or you can do it experimentally. So a group of Drosophila, or fruit fly, the little annoying fruit flies that fly around, biologists decided to test allopatry by taking a population of species of Drosophila and separating them into two populations. One that is grown on starch media and one that is grown on maltose media. So these are two different carbohydrates, right? And they only allow these Drosophila to grow on the starch or the maltose media. And you can imagine some of them are going to digest starch really well and some of them aren't. And they're the ones that don't are probably going to die. But the ones that do digest starch really well are going to breed, reproduce, right? Same with maltose. Some will do it really well. Some will die off because they don't digest maltose very well. And only the ones that can deal with maltose effectively are the ones that are going to reach reproductive age. And so let's breed these for like 60 generations, 60 to 100 generations, up to a year and a half to 3 years, depending on how long you want to breed them. And so when you do that and then bring them back, take a few of the females and males after 60 generations and put them in the same jar and let them interact and breed. Interestingly, the starch flies only want to breed with the starch flies, and the maltose flies only want to breed with the maltose flies. So that it's like 15, 16 to 1. 16 out of 17 times, the maltose picks the maltose and the starch picks the starch. And then every once in a while you'll get a random mating between a maltose and starch. And there are differences in their reproductive behaviors and who they want to breed with. So you can do it artificially with flies relatively quickly within a couple years if you keep populations separated. There are a bunch of natural examples of this, and this one is one of the best examples. I love this one. So this is the bottom of Central America, and the last part of Central America that actually rose up out of the ocean was the Isthmus of Panama and Costa Rica. And so there used to be open ocean here where the Atlantic could communicate with the Pacific, right? And so when you look at the coastal waters on either side here, there is a species of shrimp that is pretty common that looks alike, and there are 30 different species. Interestingly, 15 of them are found on the Atlantic side, and 15 of them are found on the Pacific side, which suggests to scientists, and I'm talking about right on the coast, right, at different depths. Obviously, they can't cross and interbreed because of the land mass now, which has been there for about 8 to 10 million years. What's really cool is when you look at their genetics. So when you look at their genetics, what's really interesting is that they pair up. And so if I draw like the barrier here, right? And I call this the Atlantic side and I call this the Pacific side. And this is my isthmus, right, of Panama. If you look at the species, the deepest species will pair up with the deepest species as far as the most closely related. And it goes like that all the way up so that number 1 and number 15, they pair up. This is the most recent ancestor or the most recent divergence. This is the furthest divergence. When you look at the molecular clock, these two are the oldest as far as relatedness, and these two are the youngest and the closest as far as relation goes, which tells you the isthmus or this landmass rose slowly. These got separated first, then those, and then those, and they couldn't interbreed. So it's slow progression. It took about 4 to 8 million years, and they've been separated for about 8 to 10 million years. It's pretty cool. So that suggests allopatry, right? And gives you old versus new as far as how closely related something is. There are lots of examples like that. Any questions on allopatry? And the second one is harder. The second one is called sympatry, or sympatric speciation. It's when a new species arises in a population. So think about this. Think about how hard that is to kind of even think or visualize that concept. If I separated us, this group right here of us, into a population, and then in a million years, if a new species of human arises out of this group right here. How is that possible, right? Not through reproductive isolation, but something happens within the population to give you a new species. For a long time, we knew this was a possibility, but we had no examples of it. We had no examples of it. So here is an example right here. Remember those little flies I was telling you about? They're called apple maggot flies. They look like spiders. What's interesting is that apple maggots, these apple maggot flies feed on hawthorns, which is the native apple that you find in the Midwest. I think it's in the Midwest, yeah. So this is what they feed on naturally. And you can imagine back in the 50s and 60s when they started bringing in apple trees, and growing apple trees, which aren't native. Some of them are not native. Most of them are not native to the U.S. This gave the apple maggot fly another host fruit, right? What these guys do is they lay their eggs on the apples, and then the larvae burrow into the apples, and that's what they feed on. There are a group of the apple maggot flies that prefer the hawthorns, the ancestral group, and then there's a new group that prefer the apples to feed on and the apple flowers. And that's where they meet their mates. So they meet their mates on the fruit that they feed on and lay their eggs on, so that now you have two reproductively isolated groups that are genetically diverging slowly. So this is spatial separation, right? Or what we call ecological niche specialization, where you got one preferring one food source and one preferring the other, and they're mating at their food source. Okay, so we talked about allopatric speciation last time, right? And we were just starting this. So allopatric speciation is pretty intuitive. You put a barrier, so you have a population, you separate the population into two populations by some barrier that's not passable. And then those populations spend a long time apart and they become two new species because they both accumulate these micro changes based on selection in those two new environments that prevent them from being able to breed, and then they become new species. Sympatry is a little harder. Sympatric is you have a population, there's no barrier, and then all of a sudden there are two species. So how is that possible? How is it possible to reproductively isolate a certain number within a population so that they start interbreeding but not breeding back with the original parent population? And this happens now, we know, in three different ways, at least three ways presently. We might find some new ones as we continue to explore. But for a long time, we thought this was impossible, but it's a possibility. We now know there are three different ways to do this. The first one we kind of started, I think last time, or maybe we did or didn't, and it's called polyploidy. So this is when the offspring inherits more chromosomes than the parents. And you guys already know if we have an offspring that produces chromosomes that are different than the parent, then it's not going to be able to breed back to the parent, right? And get the right number of chromosomes. So the only way it's going to be able to breed is to breed with itself or something with a similar chromosome number. And this is also very common in plants, but not in animals, right? Animals, most animals at least, don't do well with multiple sets of chromosomes. But plants don't care. So we actually do this on purpose. We do polyploidy on purpose with our agricultural plants. We actually breed plants on purpose that aren't related to get odd organisms that do things like yield more wheat or are drought resistant, those kinds of things. So a lot of agricultural science right now is trying to breed individuals and then cause mitotic error on purpose by exposing the offspring to chemicals that make them increase the number of chromosomes. Plants are okay with that, and then those outcomes of that type of breeding experiment can lead to a new species that has many sets of chromosomes. So give you an example, the wheat we use today to make our breads and all of that is a hexaploid. It has 6 sets of chromosomes. And so they've been bred artificially for hundreds of years to get us to the alloploid, which is the next type of ploidy of wheat that we use today. So what's the difference between the two types? There's one type of polyploidy called polyploidy, and then there's one that's called allopolyploidy. So what's the difference between auto and allo? Auto means same, allo means different. All right. So what do I mean by same and different? What I mean by autoploidy is the new species arose from a single species. All right, so this new species came from just one other species. So you have a parent species here, and in this instance, it is diploid, and our diploid number in our little example up here is 6, right? 6 chromosomes, or 3 homologous pairs. And you can imagine if there's a mitotic error in the germ cell that doubles the chromosome number so that you go from 2N to 4N, this germ cell in an animal will die, right, if that were to happen. But in the plant, the germ cells don't really matter, don't care how many chromosome pairs they have. So this germ cell now undergoes meiosis and becomes a 2N gamete, where it should be typically an N gamete, right? That 2N gamete meets another 2N gamete to form a new tetraploid, all right, 4 sets of chromosomes. And so how does this happen? Remember, plants can self-pollinate, right? So even though this new baby could not breed back to the adult or to the parent species, right, because it has the wrong number of chromosomes, it doesn't care. It just self-pollinates and forms its own offspring. And if other tetraploids arise this way, since it's pretty common, it could probably breed with other tetraploids, right, with those that have the right number of chromosomes. So the reason this happens is because of either mitotic or meiotic error, and in autoploidy, it typically occurs with a single species giving rise to 2 new species. So within 1 or 2 generations, you now have a completely different plant that is breeding amongst itself and is completely reproductively isolated from the original parent population. That's autoploidy. Alloploidy differs when 2 plants, two separate species that don't interbreed, produce a hybrid. That's what a hybrid is when 2 separate species breed to form offspring. It's called a hybrid. This occurs when 2 closely related plants mate. Sometimes they have different chromosome numbers, like in this instance, or other times they have the same chromosome number. Our only definition of a species is they can't interbreed, right? Normally. So they could have different chromosome numbers or the same. In this instance, notice we have species A is one plant, species B is another plant. Notice our chromosome numbers are different. So obviously they typically would not interbreed because their gametes have different numbers of chromosomes, right? But again, if the pollen were to meet an egg and you get a hybrid, this hybrid would be sterile because it has an odd number of weird chromosomes, correct? So it's sterile. It might live, but it's not going to breed. It's going to die without breeding. But if some of these hybrids were to have a mitotic or meiotic error that doubled their chromosome number, then they would have an even number of chromosomes again, right? They'd be back to 10. 10 is different than 6 or 4, and now you have a new species that can interbreed with itself and produce a completely new species and be fertile, right? Because it has an even number of chromosomes to do meiosis. All right, this is weird. Animals can't do this, but plants can. And it happens obviously more often than not. Matter of fact, they think about, if you look at the last slide, about 80% of all plant evolution probably happens because of polyploidy. Now, I'll give you an example of where alloploidy has happened. We do it on purpose in agriculture, where we breed 2 different plants that have different numbers of chromosomes or are closely related. But there are some wild examples of this. There is a genus of plant called Trogopon, which is called goat's beard. It's got really pretty flowers that bees love, and they're found mostly out west. they were brought over, or at least one or two of the species were brought over from Europe. There are actually 3 parent species that all have the same number of chromosomes, but they can't interbreed. Basically what happens when any of these 3 parent species typically interbreed, they produce a sterile offspring. I mean, even though they have the same chromosome number and pollen and egg can meet and actually produce an adult plant, the adult plant is sterile. It can't produce babies. But what happens sometimes when they do interbreed, the sterile individual undergoes a mitotic or meiotic error in the germ cells, and you double the chromosome number. So you go from 12 to 24. And the 2 hybrid species that are there have 24 chromosomes, which means now I have another set of chromosomes and I can do meiosis, right, correctly, and produce now hybrids that can interbreed or self-pollinate, and you get a couple new species of hybrids here. So, and they cannot, obviously with 24 chromosomes, breed back to the parent population. So this has happened within the last 250 years or so. These plants were brought over from Europe, and now we're starting to see hybrids that are doing quite well, actually, in certain parts of the western United States. So polyploidy is one way we can get instant speciation almost, right? The second way, and I guess I should mention, I've already mentioned this, right? That a lot of our agricultural plants, we do this artificially. And so the wheat we use today is an aloe hexaploid. Aloe meaning it was wheat species in a wild grass that were forced to breed, and they produced offspring that had a mitotic error, and now they have 6 sets of chromosomes, and this is what our typical wheat looks like now. It's an hexaploid, and produces the grain we need to make all of our yummy, right, foodstuffs, if you will. Okay, the second way we can do sympatric speciation is through sexual selection. So sexual selection, remember, means that one of the sexes is selecting for the other. Typically, it's female choice. The females are selecting males. And this can happen because of like birdsong. It can happen based on what you look like, what your color pattern is, what colors you represent. And it's pretty common in fish, particularly visual cues of the male, a certain color and pattern. And with cichlids are a type of freshwater fish you can actually buy at pet stores. They're fantastic. A group of fish that have very interesting behaviors that are fun to watch in the aquarium. Cichlids have a lot of sexual selection across the species on our planet. One wonderful example of very quick evolution that has happened with the cichlid group is the Lake Victorian cichlid groups, species that are found in Africa, which is a huge lake. And within the last, they say 150,000 years, a number of species, over 100 or so of species of cichlids, have arisen within this lake from a few parent species. And most of that has been done through, they think, sexual selection, where females start picking for color patterns and only breed with those males, which immediately reproductively isolates them, right, from all of the other fish. And here are 2 closely related species that are considered what they call sibling taxa or sister taxa within the cichlids. And you can see there, one's kind of a greenish yellow color and one is more of a purple orangish color. Females of each species tend to choose the male of their proper color pattern and very bright, clear water. Interestingly, scientists have taken these 2 species and put them in the lab and put a big, huge orange monochromic light over top of the aquarium and replacing the natural light, and so that the males all look orange, just a uniform kind of drab orange color. When that happens, females just breed with anybody, and they produce hybrids that can then breed. And so the only thing keeping these 2 species separated is female choice, right? Yep.
You mentioned that they're the hybrid bred together, but I thought you said hybrids couldn't breed together.
They can in some instances, not always. Some hybrids are sterile or some hybrids can't breed at all. But in this instance, these hybrids can.
Does that make that a new species?
Well, it might. Yeah, it's happening real time, so we don't know. What that tells you, if you can take two species and remove the sexual selection and they can interbreed, that means that they only recently diverged from one another. Recent enough that there are no pre-zygotic or probably post-zygotic barriers that prevent them from interbreeding. So they haven't been separated from one another for very long, right? As far as time goes. This is actually happening in Lake Victoria. They're starting to see a lot more of the hybrids. And the hybrid is actually a really pretty fish. It's a beautiful fish. It's kind of a combination of the two. And the reason for this, it's actually happening in nature, not only in the aquarium. The reason for this is because of human influence. Most of the land around parts of Lake Victoria in Africa have been converted to agricultural land, and they're growing crops and they're spraying fertilizers on those crops, and that fertilizers are ending up in Lake Victoria, and the water's becoming much more cloudy and polluted. And so fish are having a harder time because the water's not as clear, distinguishing between color pattern in the males. And so they're breeding pretty indiscriminately in parts of Lake Victoria now, and the hybrid is becoming pretty common. So we can have a large influence on this by causing two species that might be separated by sexual selection or might be separated by distance, right, habitat, and bringing them together so that they can produce hybrids. Then it's interesting to see if those hybrids can interbreed or not, which gives us some information about how closely related two species actually are. Okay, so we got polyploidy and we have sexual selection for sympatric populations. The last one is called heteropatry or ecological divergence. And this is our little flies we talked about a little bit yesterday. They're called apple maggot flies. So they are found throughout the United States. They even range down up into Canada and down into Mexico a little bit. And there are several species of them. What's most interesting about them, at least in my opinion, is that they mimic jumping spiders. And they do that by having a color pattern on their dorsal or back that looks like the body of the spider, and then their wings have feet or legs painted on them that look like little jumping spiders. And then they even move and jerk around like a jumping spider does, so that they're mimicking a spider so that predators don't think they're a fly. They leave them alone, right? Because spiders can eat other insects. But they're called apple maggot flies because they actually lay their eggs and the maggots feed on apples. So originally, the original populations of these apple maggot flies before apples came to the US fed on this plant, which is called hawthorn, which produces a fruit that's similar to apples. The big thing is it's much slower in its development, takes longer to ripe, and is not as common as apple trees now in certain parts of the US where we grow apples on purpose. So about 250 years ago, we brought apples to the US, right? And we grow them agriculturally now. And of course, these flies, at least part of the population, started feeding and laying eggs and reproducing on the apples versus the hawthorns. Now, interestingly, apples, we have grown them artificially so they produce big fruit and they do it quickly. Apple trees grow apples relatively quickly, which means that this food source is readily available in certain parts of the US, that these flies are now staying either on the apples or on the hawthorns, and they are becoming habitat separated, right? That pre-zygotic barrier that we consider habitat separation or isolation. And that's happened for the last 200 years or so. And now scientists are seeing some major differences in the genotypes and phenotypic behavior of these apple fly maggots. For example, The population of apple fly that likes to hang out on apples and breed, their development has been sped up considerably compared to the original population, which is going to inhabit, feed, and lay their eggs on the hawthorn. Hawthorn has slower fruit development time, and so your development needs to be slower. Whereas the apples, their development time is faster, the fruit development time is faster, and so the flies' development time is sped up. Interestingly, if you take hawthorn flies and make them eat or feed on apples and lay eggs on apples, the larvae don't do as well, and vice versa. And so you're starting not only to get habitat isolation, but you're getting temporal isolation, where there's this timing feature to development. And if the alleles continue to change, eventually you're going to get probably some hybrid viability issues or some hybrid sterile issues that lead to post-zygotic modifications that might lead these guys to be separate species. Right now, the species that live on apples versus the species that live on hawthorns are only considered subspecies of each other, not true separate species. But over the next 1,000 years, if this continues, or the next 250 years if it continues, you might have two separate species that don't interbreed at all. And that's called ecological divergence. And this is a way evolution happens a lot, probably, particularly evolution driven by natural selection, because selection is going to lead different individuals within a population with genetic variability to occupy certain niches and then stay in those niches. And niche isolation can lead to reproductive isolation. Does that make sense? And that is called heteropatry, and is a good kind of example of how evolution can cause speciation. Now, as is already mentioned, hybrids do form, and we want to know, well, what does that mean? Is a hybrid a new species? Is the hybrids are they fertile, or are they sterile? Do they die before they reproduce? That's a big question that biologists like to answer when hybrid zones do exist. And hybrid zones do pop up between closely related species when they meet each other. That is, there are accidental breeding zones between different species if they're closely related. So for biologists, this is with our influence on the planet, particularly with global warming. Global warming is allowing species that used to be occupied just the southern United States actually move north, right? Because it's staying warmer longer in northern states. Great example of this is the southern. Have you guys ever seen flying squirrels? The squirrels that fly. There's a southern species and a northern species. Northern species lives here in Indiana. I have them in my backyard. They're pretty cool. But the southern species is creeping slowly north, and the southern edge of the northern flying squirrel and the northern edge of the southern flying squirrel are now meeting, and there's a hybrid zone now. They never used to overlap. There used to be hundreds of miles between the two populations. But now, over the last 5 or 6 years, there is a hybrid zone. Those hybrids are surviving and growing up. Right? We have no idea how successful they are reproductively yet, because it's pretty new. But it'll be interesting to study that hybrid zone, right? And see what happens. So when hybrid zones do jump up, we want to know, are the hybrids able to compete, right? Are they able to survive? Are they able to reproduce? Are they sterile? Are they not? Sometimes hybrids are more viable than the actual parent species. It does happen. But often the hybrid is not as viable as the two parent species and die off before they can reproduce, are born sterile, or we have no idea at this point. Another great example of we have no idea what's going to happen is this right here. Right? These two bears are the results of grizzly and polar bears mating, and they're called growler bears. These guys are starting to pop up all over the range of where polar bears and grizzlies meet, because that hybrid zone has become more common over the last, say, 10 years because of global warming, has melted ice caps or is melting ice faster than it should during seasons when ice should be there up in the Arctic. So I don't know if you know much about polar bears, but polar bears need ice in order to survive. They need ice to hunt. They need ice to rest. They're great swimmers, but they typically ambush hunt seals on ice. If there is no ice, then there's going to be no seals. They're going to move, right? And so what happens with polar bears when they can't find food is they head inland, right, searching for something to eat. And that's when they meet the grizzly bear, right, which is a terrestrial bear. And when they do meet, grizzlies and polar bears are close enough related that they reproduce. They actually breed. And when they breed, they produce this intermediate bear called a Growlar bear. And this bear actually can grow up and reproduce on its own. So recent studies in the early 2020s, have shown that the growlers, females, are breeding with grizzlies and polar bears, producing F2 and F3 generations. And those bears are growing up and surviving. And so we're not sure what the result of this is going to be. Most biologists fear that polar bears are going to disappear, right? Because the ice is going to disappear. And if they're breeding with grizzlies, you're going to dilute out the polar bear traits, right? If you look at this guy right here, this particular Growlar bear looks more like a polar bear, but it doesn't look like a polar bear, right? It's shorter. Its neck is much shorter. Its feet are too small. It's not a good swimmer like a polar bear is. I don't know if you guys know, but polar bears are great swimmers. They can swim days in open ocean, no problem. Because of their long necks, they can be above the water, and because of their large feet, act like paddles. And so the intermediate hybrid is going to carry traits of both grizzlies and polar bears. And the outcome of this, we're not sure of, right? We'll just have to keep watching the hybrid zone. Now, if you do get a hybrid zone, there are 3 possibilities to our speciation here, right? Can we reinforce the speciation? Are we going to weaken the speciation like maybe this is weakening, right? The separation of the 2 species. Sometimes if 2 species meet and they produce hybrids, all the hybrids are sterile, right? That just reinforces the separation, correct? Or you can get kind of what we call stabilizing type of zone or stabilizing features, and I'm going to go over the 3 of these. So hybrid outcomes, there are kind of 3 possibilities of hybrid outcomes, and these are the 3 right here. All right, these are kind of drawings that represent them. And I like the drawing because I'm visual, right? I need something to look at in order to understand this. So here we have 1 species in 3 different populations. It could be one of these birds, for example, down here, these flycatchers, or the polar bear, for example, right? And with these populations here, you notice that the ancestral setup was that the 3 populations could interbreed. That is, some of the individuals from this population could move over here and breed with those guys and so forth. And so this is called migration. There was always genes migrating in and out of these 3 populations. A species doesn't have to be found in a single population. It can be in multiple populations, right? Across a geographic range. Now, if something happens here to form a barrier to gene flow, a mountain range, or some sympatric sexual selection event, right? That happens, for example, in birds, like these guys down here, would separate one of these populations from the other 2. And now this population here, the 3rd one, is on its own trajectory of natural selection, and these 2 populations are on their own trajectory of selection. And let's say they're separated for whatever reason for, 1,000 years, 2,000 years, 100,000 years. If for whatever reason they come back in contact with one another after a certain amount of time, what happens to the hybrids, right, is the question. So you got 3 possible outcomes of this new hybridization, bringing the species back together after a certain amount of time. The first one's called reinforcement, in that I'm reinforcing the idea that, hey, I'm separate species. The hybrid is going to be not viable, it's going to die, it's going to be vulnerable, it's not going to reach reproductive age. there's going to be nice prezygotic or prozygotic barriers that prevent the two from forming a viable hybrid. So reinforcement, a good example of reinforcement, it happens in sympatric species. All right, is a good place to look for it. A great example of this, of reinforcement, is when you compare birds in particular, where you have a population of flycatcher that is sympatric and a population of flycatcher that's allopatric. So we have two species here. We have the pied flycatcher and the collared flycatcher. Now, what's interesting there are some populations that have developed separated from one another by some barrier, whether it's forest or a river or a mountain range. And when that happens, you have two allopatric populations. Interestingly, when you look at the allopatric populations that have been separated for thousands of years, the male of the pied looks exactly like the male of the collared. And so that when you bring females together to choose, they don't know which one to choose, right? So allopatry doesn't care much about sexual selection in birds because the populations have been separated. They never run into each other. Whereas in a sympatric situation, which shows really strong reinforcement, when the pied species is found in the same area as the collared species, the males look very different from one another. Not only do they look very different from one another, their song is different. Therefore, females of pied only pick pied, and females of the collared only pick collared, because sexual selection is keeping the hybrids from happening, right? Which is prezygotic. And so it reinforces this idea of sexual selection being a really good speciation tactic as far as producing and reinforcing two new species. So as these guys continue to be separated from one another, you can imagine the further and further out in time you go, the harder and harder and harder it'll be able to get a hybrid, right? And that would be reinforcing. The opposite of that would be to bring them back together, right? So they go up, they're separated, you bring them back, and then all of the hybrids are either stronger than the actual two parent populations, or the hybrids can compete with the two parent populations. That's called fusion. And so it'll look like this, right? You get your separation, you bring them back, and then over time, you're going to have just one species again, right? And not two. The other one is called stabilizing, where hybrids arise a lot, but they never seem to be able to make it, and the parent populations are going to be separated indefinitely as long as the stabilizing environment remains. So let's look at two examples of that represent fusion and a stable hybrid outcome. So stabilization happens when you get a common hybrid zone and the hybrids never make it. All right, they never make it for a number of reasons. One of the most interesting hybrid zones between two closely related species happens in the fire-bellied toad. So the fire-bellied toad is the guy you got down in the lobby down there, the little frogs that are in the tanks. They're very, very popular because of their red bellies, and they're easy to breed. These guys are really easy to breed. But in the wild, the red-bellied toad or fire-bellied toad is separated from another parent species, closely related species, that's called the yellow-bellied toad. And the yellow-bellied toad is found in Europe, as you can see here, in yellow. And then the fire-belly is found mostly in Indonesia, in Asia, and those areas of the world. And there's a hybrid zone between the two parent species, the orange and the yellow, that runs along this red line. What's most interesting about this red line is for 4,000 miles, the hybrid zone is the same width, 10 kilometers. It's crazy. There's no widening in some places and shrinking in others. The two parent species are completely separated from one another, and where they do meet, they will breed, And then the hybrid zone is really small because the hybrids don't do well. They just die or are not adapted to the environment. So there are two things that drive this stable hybrid zone. That's why it's called stabilization. First and foremost, the hybrid dies. Its viability is very low. Second, the second thing is the actual habitat that the hybrid zone is found in. So the fire-bellied toad, this guy here in orange, it lives in lowlands, low elevation. Okay? The yellow-bellied toad lives at high elevation. So one lives at high and one lives at low. The hybrid zone meets in the middle. It's where the parent species are sneaking, the fire-bellied sneaking up and the yellow-bellied sneaking down. They obviously do either well at high elevation or low. In a middle elevation, neither parent species does well. So they're very niche specific as far as elevation goes. And the hybrid obviously doesn't do well at all. And so this keeps the zone at this middle elevation the same width across the entire range of the two parental species. Pretty cool. Very interesting. So that's called stabilization. So here we have habitat and actual poor hybrid viability that are maintaining a stable hybrid zone between the two parent species. Make sense? Okay, the last one is called fusion. And fusion happens when you have separation of two species, but they haven't been separated long enough, and it's going to reverse your speciation event. So you have one species giving rise to two, and then when you bring the two back, they can interbreed, and all of a sudden you only have one species again. So this is called weakening of the reproductive barriers. Basically, when fusion happens, when you bring two species back, it simply means they haven't been separated long enough, right? In order to get proper post-zygotic barriers to have formed, there's not enough change genetically. And this is what's happening with your cichlids in Africa. When you have two species that are simply separated by sexual selection, when you remove the sexual selection factor, you produce a hybrid, and I think the hybrid's really pretty between the two adults. And these hybrids are doing fantastic in Lake Victoria. They can breed, and I think there are even a few studies that recently that suggest they can outcompete the two parental species. And so you can imagine maybe 10,000 years from now, there will be no more separation of the two species. You'll only have the hybrids. You only have one species, right, instead of two. That will definitely happen if the reproductive barriers continue to weaken over time.
So is the new hybrid, is that like its own species, or is it just like a subspecies?
Right now, it's just a hybrid.
Oh.
Right, and so because these two are still considered separate species, the hybrid isn't considered anything but a hybrid for now. Over time, if these guys disappear, then it will become, the residing species.
So like, because it has like the species name next to the words of like Pendamillion, and it just has nothing next to it. So you would just call it a hybrid.
Yeah, we call it a hybrid. Yeah, they wouldn't give it a species name. Yep,
Okay.
you got it. All right, the last thing we can cover, at least with this chapter, which is chapter 24, is just speed, or what I call the mode of speciation. That is, how fast does evolution produce new species? And the range is anywhere from 4,000 to over 40 million years to get true separate speciation between two species that won't interbreed. So it can be really fast or really slow, right? And then there's some in between. If you take the average number across the fossil record, which has been done a couple of different times by different groups, it's about 6 to 8 million years. It usually takes about 6 to 8 million years to get true speciation where hybrids don't survive. Now, obviously, we can do that really fast with plants, even faster than 4,000 years, or we can do it much slower. So there are kind of two speeds or modes, really, to evolution that causes speciation. The gradual model is what we call gradualism, or what we call Darwinian evolution. Darwin is who, in his Origin of Species, described slow, gradual change that takes a long time, right? And that's how a lot of evolution goes. You can look at a fossil record and see that it took 100 million years to go from this to 5 or 6 different transitional fossils before you get to what's alive today, or what went extinct as the last organism in a line of organisms. So this gradual model, there are lots of examples of it across the fossil record. Punctuated equilibrium was introduced by a fellow called, his name's Stephen Gould. He's dead now. He was a Harvard professor who suggested, as he studied the fossil record, that he could find events in geologic time where there was this huge, very fast diversification of organisms, and the evolution time was very short. And so you had these periods within geologic time, or the life history of the Earth, that showed rapid rates of evolution and species diversification across the planet. So you would have these bursts of evolution, and then nothing happens for 10 million years. Burst of evolution, and nothing happens for 10 million years. That's what punctuated equilibrium is. And for a long time after Gould described this mechanism, there were 2 schools of thought, right? That there was punctuated equilibrium and gradualism. Well, like most things in biology, as you ponder things, they probably both happen, right? Gradualism is kind of the default model in my mind, and punctuated equilibrium happens when the conditions are right. So when would the conditions be right for punctuated equilibrium? Well, how about a mass extinction event, right? And I'll give you a good example. 65 million years ago, the planet was ruled by the most derived dinosaurs, right? There were dinosaurs everywhere. Mammals were here, they were really small and not very diverse. Asteroid hits the planet, destroys the environmental conditions for dinosaurs, and all of them die out. They just disappear, opening up a bunch of niches, right? All of a sudden, there aren't any dinosaurs anymore that are filling all the niches that dinosaurs filled. These little mammals now diversify, and we are now at the climax, or the middle of that mammalian diversification. Mammals rule the planet now, right? And fill a lot of the niches, big niches in particular. And the reason for that is because of the start of a punctuated equilibrium event that happened 65 million years ago. There are lots of examples of this. Flowering plants are the dominant plant. They weren't always the dominant plant. The reason they dominated is because of the symbiotic relationship between them and insects. When that happened, there was a burst of diversification or punctuated equilibrium event for flowering plants and insects, who basically need each other to survive. So there are a bunch of examples of this, these different modes or speeds of evolution that happen in the real world. So here's kind of a picture of what that looks like. So butterfly, bam, explosion of a whole bunch of new species of butterfly, and then nothing happens for a long period of time. And then you might get another explosion event where you get a bunch of different species of butterfly, and so forth. Whereas gradualism works slow. This transition, this transition, this transition, this is what you see. So you can see all these intermediate fossils that lead up to what you see today, or what is the end or climax of evolution in a line that's extinct. All right.